Hello, my name is Stuart Strand. I'm a research professor at the University of Washington. Today, I want to give you a short lecture on a new idea on how to prevent nitrous oxide emissions from agriculture. Global warming is a dire threat to human civilization. We must achieve a carbon neutral economy, but we must also eliminate the emissions of other greenhouse gases, such as nitrous oxide, N2O. N2O is a greenhouse gas that's 300 times more potent than carbon dioxide. Nitrous oxide is also the main threat to the ozone layer, which protects people and ecosystems from the deadly effects of ultraviolet radiation. Most of humanity's nitrous oxide emissions come from agricultural soils. Nitrogen fertilization increases nitrous oxide emissions, and nitrous oxide is the main air pollutant emitted by plant agriculture. Existing attempts to reduce nitrous oxide emissions often result in decreased crop yields. And these nitrous oxide emissions come from organic fields just as much, or if not more so, than they come from fields with conventional fertilization. If nitrous oxide emissions from fertilized agricultural soils were brought to zero, then food production could be uncoupled from nitrous oxide emissions. So we're proposing a new genetically engineered technology that can prevent nitrous oxide emissions from crop soils. In the soil, bacteria can use the enzyme nitrous oxide reductase to turn nitrous oxide into harmless N2, but in most agricultural soils, this enzyme is at least partially turned off, and thus the nitrous oxide is emitted. We could come up with genetically engineered bacterial strains that constitutively express nitrous oxide reductase, that is, has nitrous oxide reductase turned on all the time. But when engineered bacteria are introduced into soils, they rapidly die out. So instead, we will introduce bacterial nitrous oxide reductase into the mitochondria of crop plants. Now, mitochondria are the powerhouses of eukaryotic cells, whether plant or animal. It's where ATP is made, and there are lots of mitochondria in eukaryotic cells. Bacteria and mitochondria derive from a common evolutionary ancestor. The mitochondrial inner membrane space is analogous to the bacterial periplasm. So here we can see an illustration of a bacterium down here, which has a double outer membrane. And between those two membranes, there is the periplasmic space. And that's where the nitrous oxide reductase does its function. And in mitochondria, you see the same sort of thing. You see a double membrane on the outside of the mitochondria, and between those two spaces is the inner membrane space. And that's where we're going to put the nitrous oxide reductase in our transgenic plants. We propose to use the mitochondria as surrogate bacteria for modification with nitrous oxide reductase. Mitochondrial proteins can be modified by traditional plant transformation. 99% of the proteins in mitochondria are coded on the plant nuclear chromosome, where we commonly can modify plant protein expression. These proteins are translocated by targeting signals on the protein that act like the addresses or zip codes on a letter. And many experiments have demonstrated that the correct translocation to the mitochondria of genetically engineered proteins containing these targeting sequences. We will introduce nitrous oxide reductase into the electron transport system of the eukaryotic mitochondria, as in the same locations as they are in bacteria. Previous experiments have identified which helper proteins are necessary for assembly of the nitrous oxide reductase. And here you can see a schematic, and green and purple are the two components of the nitrous oxide reductase, the NOS-Zs, and then the L, D, Y, F, R, and X are accessory proteins that are necessary for the proper functioning of the nitrous oxide reductase. And this is in the bacterium, but we're going to take the whole system and put it into the inner membrane space of the mitochondria in plants. So how much good is this going to do? And we can analyze the root uptake mathematically of nitrous oxide by these transgenic plants, assuming that we can get this to work in plants. The flux of nitrous oxide into a plant root expressing nitrous oxide reductase can be modeled by equating the nitrous oxide reductase removal rate to the diffusion rate of nitrous oxide from the bulk soil where nitrous oxide is made to the root surface. And here we have some equations that outline the process of the derivation of this formula for the flux. And this flux is given as a function of all sorts of parameters that are here, but all of these parameters are empirically observable and have values that have been determined for them. So if we plug all of those into this equation, we can get a flux to the roots, and the potential flux from the bulk soil where nitrous oxide is made to the transgenic roots where it's consumed is estimated to be about 30 kilograms of nitrous oxide per hectare, that's a unit of crop area, per year, which is about 3 times greater than the observed average emissions of nitrous oxide from fertilized cornfields, which is about 9 kilograms of nitrous oxide per hectare per year. So we should be able to completely stop these emissions. So emissions from cornfields could be reduced to 0, avoiding on average about 9 kilograms of nitrous oxide emissions per hectare per year. In addition, the above ground parts of the transgenic crops could remove nitrous oxide from the atmosphere through the stomata of their leaves. Plant leaves are efficient absorbers of trace gases. They have evolved stomata in their leaves, which open to admit carbon dioxide into their leaves, where photosynthesis converts that carbon to their structure compounds, sugars, and so on in the plant. Nitrous oxide could also enter into the plant leaf interior through the stomata, where it could be acted upon by the nitrous oxide reductase in the mitochondria and converted to harmless N2. So to model this process, we set up this formula for the mass transport flux equal to the enzymatic flux, and using mass transfer coefficients that have been observed for CO2 and corrected for the diffusivity of N2O, we can come up with a flux S formula like this. And all of these parameters are observable. And so we can calculate a flux from the atmosphere to a field of transgenic crops expressing nitrous oxide reductase. That would be potentially about 5 kilograms uptake from the atmosphere per hectare per year. So would nitrous oxide reductase activity in transgenic crop plants significantly affect atmospheric N2O concentrations? Well, nitrous oxide reductase in roots and leaves, if we can make it work, would prevent the emissions and remove from the atmosphere about 14 kilograms of nitrous oxide per hectare per year. So what acreage, what area of plants would be necessary to prevent the annual increases in nitrous oxide? Well, nitrous oxide concentrations in the atmosphere are shown here historically from 1985 to the present day, going up from around 304 parts per million, parts per billion, to where it is now at above 330 parts per billion. And that rate of increase is about 1 part per billion per year, shown here. So if I take 1 part per billion and I consider the entire mass of the atmosphere, that means that nitrous oxide is increasing in the atmosphere by about 8 teragrams of nitrous oxide per year. A teragram is 1,000,000 tons. So if I take 8 teragrams and I divide it by 14 kilograms, I end up with an estimate of the acreage necessary, the area necessary for these crops, to prevent the annual increase of nitrous oxide would be about 500 million hectares of nitrous oxide reductase expressing crops, about a third of the total cropland on Earth. Importantly, the cost of this technology would be low. Once the nitrous oxide reductase expressing plants were created and producing seed, these plants could be easily propagated and seed production scaled up with nearly no additional cost over conventional seed. Farmers would be able to claim carbon credits on the carbon market of about $60 per hectare per year with current carbon market prices, thus incentivizing planting of nitrous oxide reductase crops. In conclusion, the proper location for nitrous oxide reductase in engineered eukaryotes is in the mitochondria. Nitrous oxide reductase plant technology could potentially prevent crop field emissions and directly remove nitrous oxide from the atmosphere on a global scale. This technology by itself, sustainably and at little cost, could potentially stop increasing nitrous oxide in the atmosphere, removing the major source of atmospheric pollution from plant agriculture. Well, if you like these ideas and want to help us make them work, please visit our lab website shown here and down below and click the donations button. While you're there, you can also learn about our genetically engineered houseplants that clean your home's air. Thank you.